Hey guys, today we are going to talk about gene expression and get started with understanding the basics. So gene expression, as we'll talk about, is going to encompass both transcription and translation. This first part will only be talking about transcription and related RNA processing. In your second video, we'll get more into translation. So what is gene expression? All of the cells in an organism, including you, actually contain the exact same DNA. So every cell in your body has the same genetic code. But we've talked about in previous chapters, and it's pretty obvious if you even just think about the way a human body is going to be composed, that different cell types have different properties. Muscle cells look different from liver cells, which look different from neurons. So if they all have the same genetic code, what makes these cells different from one another? So it turns out that this is all the result of selective gene expression. So even though cells have the same set of DNA, they can turn some genes on and turn others off, depending on what they're supposed to be doing or in response to signals from the environment or even their own internal signaling. So they're basically sensing the environment and using the DNA and deciding, am I going to make a copy of this as mRNA, or am I going to try and prevent a copy of RNA from being made? So gene expression, when we talk about it as scientists, this is simply the generation of what we refer to as a gene product. So a gene product can either be measured as RNA, like a messenger RNA, or as protein levels. So in chapter 18, we'll be talking about some cases where not everything is made into a protein. So sometimes it's more suitable to look at the RNA levels, and sometimes it's more suitable to look at the protein levels. However, one thing it's never suitable to do is consider gene expression as what's happening in the DNA. So gene expression is not DNA. DNA can contain genes, but when we talk about expressing them, we're referring to whether we're going to be undergoing transcription, translation, not what's actually in the DNA sequence. So again, there are two basic steps to this. Transcription, which is where we create an RNA copy, we synthesize mRNA. In general, we'll be talking about messenger RNA as a copy of the DNA. So here in eukaryotic cells, we'll be talking about this happening in the nucleus and then moving out to the cytosol where ribosomes can make proteins. The second step of this, of course, if we're dealing with protein coding genes, is going to be translation. So protein synthesis from messenger RNA. Again, in this video, we're only going to be covering what happens to the RNA before it's translated. So it will be really important to make sure that we understand fully the basics of transcription, because in chapter 18, we'll be adding on to it to learn more about how regulation of gene expression can happen. So today, what we're going to be doing is learning the truth about transcription. So there are some things that you probably already learned about transcription and translation. So for some parts of this, we learned that prokaryotic cells don't have a nucleus, so transcription and translation occur simultaneously. Whereas in eukaryotic cells, this is going to occur in the nucleus. So that means transcription happening in the nucleus, we have a chance to process RNA, which isn't available to our prokaryotic friends. After we exit the nucleus, we can undergo translation. So all of those things that you've learned are true. Making an RNA copy of our DNA is true, but it's a little more complicated than your textbooks have probably made it out to be to begin with. So we'll start with what we do know. Transcription is the process of making an RNA copy of DNA. So again, the way to think about this is our DNA is going to contain the master set of information. The RNA will contain a copy of that information that is going to be a little bit transient and expendable. So our DNA is a permanent thing. This would be like a permanent copy of something. Let's say you have like a master Excel spreadsheet, and whenever you want to edit it, you make a copy of it and you can edit away. We're not going to really be editing the RNA too much right now, but just understanding that our DNA is permanent. whereas our RNA is going to allow us to make a copy of that we can use for other purposes. So RNA, or ribonucleic acid, has a backbone made of ribose sugar instead of deoxyribose. So still have those phosphodiester bonds, but we're using a different sugar. So this is ribose, and this is deoxyribose. And they're very, very similar, except for ribose has an extra hydroxyl group coming off of carbon 2 here. Okay, so our deoxyribose, so named because it's got one less oxygen here, deoxy, and then the sugar's ribose. Okay, so the reason that's important to note is because this extra hydroxyl group makes the RNA more delicate than DNA. So it really has sort of the tendency to degrade and fall apart in aqueous solution, which is found in our bodies, it's found everywhere. So really this degrades very quickly if it's not given some help to stabilize. So that's both good and bad. In the cell, it's important to remember that RNA is gene expression. It's turning a gene on or off. If we're reacting to something in the environment or signals that the cell gets, it's kind of important that RNA be made only in the case when we need it. So RNA really doesn't need to hang around after its purpose is complete. So this extra hydroxyl group gives RNA more of a transient existence, which is the way that it's supposed to be when compared to DNA, which is our permanent copy. So again, this transience in our cells makes it really useful for responding to the environment. It does make it a bit tricky to work with in the lab, and this is part of the reason that it's so difficult to get some definitive answers on some of those COVID tests that people have been taking. Okay, can contribute, but the stability of RNA is just a little bit difficult to work with. Okay, so there are other reasons contributing to some of those COVID testing issues, but that's definitely one issue that contributes. Okay, so we know a little bit more about RNA. So now let's talk about making this DNA copy that we're talking about. So RNA is synthesized by an enzyme called RNA polymerase. So this is important, not DNA polymerase. These are pretty easy to remember actually, because DNA polymerase makes DNA. So that's used in replication. This is not replication, okay? Replication only happens when cells divide. Transcription happens continuously all the time. So RNA is synthesized by RNA polymerase, so it's easy to remember because it simply tells you the name of the molecule that it's making. So RNA polymerase will use one of the DNA strands as a template. Okay, so this is called the template strand, and we're going to just create an RNA strand. We're going to add on RNA nucleotides or ribonucleotides using base pairing rules. So one difference being, however, that instead of using thymine to pair with adenine, we're going to be using uracil. So no thymine to be found in our RNA, but otherwise these base pairing rules are very similar. Okay, so using those base pairing rules, synthesis of the RNA molecule occurs just like DNA synthesis by only adding to the 3' end. So this again will be synthesized in the 5' to 3' direction. Okay, we're still anti-parallel. Okay, so note the strand that we're working on here. The 5' end is over here on the right. Okay, here our 5' end is on the left. So we're still anti-parallel and synthesizing in the 5' to 3' direction. So it's important to note again that at this time we're only using one of the 2 strands. So the strand that we're actually using to pair with RNA is called the template strand. The other strand, here it's labeled as the non-template strand, but you may also hear this referred to as the coding strand. Okay, so it's important to realize which one you're dealing with at which time. If you're asked some questions about what the RNA sequence would look like, it's important to know whether you're dealing with the template strand straight off or the non-template strand, okay, or the coding strand. So just getting a little bit oriented there. Okay, so let's now talk about the anatomy of a gene. So what it even means to be a gene. So we kind of think of a gene as a unit that's going to give us some type of information or that's going to be transcribed. So anything that's going to be transcribed, we have to mark it as an area that needs to be transcribed. Okay, there's a lot of DNA in your genome. Some of it is going to be genes that code for a protein or some other RNA product. Some of it is going to be regulatory sequence. We'll see some of that today, and you'll see some of that when you look at the pre-lab for next week. There's also DNA that we really don't know what it's doing there yet, but maybe it has some purpose. Maybe it's just some evolutionary leftover. Okay, so what we're dealing with a gene is going to be something that we know to be transcribed. So we have to mark this as such and get it ready for transcription. So here you can see some things labeled. So in green, we have what we call the core promoter. So this should be very close in proximity to the actual start point where transcription will begin. So the promoter is the landing site, the place where RNA polymerase will initially sit before it starts transcription. So the promoter is going to be composed of a specific DNA sequence. So the promoter is DNA. It's a series of DNA sequence. And in eukaryotic cells, this is called the Tata box or Tata box, however you have heard it said before. The important thing is TATA. So in eukaryotes, we're always going to see this Tata box. This is the core promoter. It's essential that this be here for transcription to begin. In prokaryotes, so the terminology is a little bit different. The promoter can also be called kind of the -10, -35. And what we're talking about here is from the start point, 10 nucleotides back, and then another 35 nucleotides back. Okay, and there's some sequences that are similar in purpose and structure to the Tata box, but in prokaryotes, we call this the Pribnow box. So this will be the terminology that we're looking at. Okay, so next, this blue part. So this is the part of the DNA that will actually be transcribed to create an RNA molecule. So none of the stuff that's green will actually make it into the chance to be a protein. So the blue part, this is also called a coding region. So it's the part we're going to code for, we're going to transcribe, or you sometimes hear this referred to specifically in kind of a genomic context as an open reading frame or an ORF. Okay, so those are some terminology that you may hear. This is what's going to be made. For now, we're talking about protein coding genes, so this could be a protein. Over here, this DNA, we don't know what it's doing, but it's unrelated to what we're talking about for now. It's just kind of the setup, the lead-in for here's a promoter. It's distinct from whatever's happening here. And again, in chapter 18, we'll learn about some regulatory sequences that can be found upstream, downstream, all kinds of places. Okay, so for transcription to begin, RNA polymerase must sit on the promoter. Okay, so it must sit on the promoter and it must begin synthesizing RNA. So RNA polymerase, even though it must sit on the promoter, actually has no biochemistry that allows it to find this Tata box sequence on its own. So that means it's going to need a little help. Okay, so we're going to talk about two examples, one from prokaryotes, which is the more simple example, and the second one is going to be for eukaryotes, which is often the one we tend to care more about on the front end. So let's talk about some prokaryotic transcription. Okay, so here what we're looking at is RNA polymerase, which is going to be here in red, and then what we're going to call the sigma subunit in blue. So RNA polymerase and the sigma subunit kind of hang around, and RNA polymerase again has no ability to bind the promoter itself. Okay, so if it was on its own, it really can't find anything. All right, but the sigma subunit can help it find the promoter. It has an ability to attach to a specific DNA sequence. So RNA polymerase will then open up our DNA here, our double strand. We also call this melting, and it's a little bit similar to what helicase does for our replication. Again, just to reiterate, this is not replication. This is transcription. So those are two different processes formed for two different reasons. There's just some biochemistry that's similar between the two. So our sigma subunit helped us find the location that we were looking at. RNA polymerase in prokaryotes can now do the job on its own. It starts to lay down a few nucleotides. It doesn't require a primer because this is RNA polymerase, so it is fine. And it starts putting down nucleotides, creating an RNA molecule. And once we're going to kind of get these initial nucleotides added, these ribonucleotides, then our sigma subunit can leave and this RNA can really truck along. So once our sigma subunit goes, we've kind of primed this up, we're ready to go. This is called promoter clearance. So once we're kind of off the promoter. So if we were to find the promoter here, okay, we would really be thinking about the part that's not going to be incorporated with this mRNA. It's going to be something that allows us to find the start site of our genes. Okay, so this fulfilled our two goals. RNA polymerase made it to the promoter and then it started polymerizing. So let's see how this works in eukaryotes. So in eukaryotes, the goals are the same. We got to find our promoter and we have to start synthesizing RNA. So getting there, however, while the goals are similar, we've got a couple more things to deal with. Okay, so here's our gene setup. Again, you can see we've got our TATA box, our core promoter here, our coding region. This is our start site. So to find the TATA box, that's our first goal. So again, RNA polymerase has no way to find the TATA box on its own. Therefore, we need some help from proteins called general transcription factors. Okay, so a transcription factor is pretty much what it sounds like. It's going to help control the process of making RNA. So most of the time, these are able to bind to DNA or either indirectly or directly. They're involved with transcription somehow. General transcription factors make it sound like this could be any old transcription factor, but that is not the case. These are called general transcription factors because they're going to be present every time transcription happens, regardless of any other way that we're trying to regulate it. So they're more called general transcription factors because they're the baseline case. Okay, they're always present in a eukaryotic cell. So here's our RNA polymerase along with our general transcription factors. Again, all of these are present every time transcription occurs. Okay, the exception, this is RNA polymerase 2, which is going to be largely involved with creating messenger RNA, but in eukaryotic cells, there are a couple of other types involved with synthesis of different RNA types. So the RNA polymerase in eukaryotic cells can switch out, but otherwise, what we're dealing with here, TF2D, B, TF2A, all of these guys are going to be there every time. So TF, this is transcription factor 2D, all of these transcription factor 2 followed by a letter. Okay, so again, all of these components have to be there every single time transcription occurs. And this is where we're learning the truth about transcription, because up until this point, most textbooks have really just focused on RNA polymerase just being there naked. Eukaryotic cells, simply not true. So what we're going to be focusing on in chapter 18 is going to be how do we make a promoter more attractive for these factors? So that will be where we're going with this. Okay, let's get started and see the order of this process. So again, RNA polymerase itself cannot find the TATA box on its own. It's right here, but it can't see it, okay? So we're going to need some help. the first thing that happens is TF2D will bind our TATA box. Okay, so TF2D is associated with kind of a complex that's called the TATA binding subunit, TATA binding protein. Okay, TATA binding domain, however you may hear it. Okay, and so its main goal is to seek out the TATA box. And that's what it's going to do. Of these subunits, of these components, this is the only one, TF2D is the only one that has any ability to find the TATA box. Okay, everything else is dependent on this initial action. Okay, so you can think of the D as dependent, TF2D. I actually don't recall why these are named, A, B, D, whatever, but if D helps you remember it's everything else is dependent on it, then let's go with that for now. Okay, so we've got our TF2D there. That's going to mark the TATA box, the place we're all going to bind. All right, so the next thing that happened, TF2A and B help to form this complex. Okay, and partially they're there to help stabilize this, but we're going to see some uses for B here in a second. Okay, so still getting ready. Our first goal's not been met yet. RNA polymerase is still floating out here in space, but we're getting close. So step 3, TF2F, which has been palling around with RNA polymerase 2 this whole time, they've been hanging out together. TF2F is going to kind of escort RNA polymerase 2 to the growing complex. Okay, so the reason that's going to happen is because of this association between B and F. Okay, you can think of this as like best friends. Okay, so step 1, we were dependent on TF2D to find the TATA box. A and B came in together. That's kind of alphabetical, so that's fine. Now, best friends. We've got RNA polymerase 2 associated, but we're here. Goal 1 has been met. Okay, so check it out. It's in position. You can see we're getting ready to get on this start site. All right, so you can see we've got a couple more things to go here. We've not met our second goal yet, which is going to be getting RNA polymerase to actually start transcribing. So our next step, TF2E and H join, and that's going to be where we start to really get going. So these are going to help with a couple of things, okay? First, we've completed what we call the pre-initiation complex. So we haven't initiated anything yet, but everybody's here. The party is complete, it's like ready to go, okay? But we're gonna have to get it started, and our H and E are gonna help us get there. So let's look at this. We've got our pre-initiation complex. We're gonna use a little bit of ATP, and transcription will begin, okay? So our RNA polymerase will get phosphorylated. Okay, so when you see ATP being used in a lot of scenarios, and we're kind of trained to think ATP is energy, okay, but it's not raw energy in like the form of Dr. Manhattan, okay? It's just ATP, it's adenosine triphosphate. Most of the time when we think about energy being used, it's because we've transferred those phosphates to something else, okay? We have to go get more phosphates. And here we go. So you can imagine how important phosphorylation is just in general, because ATP is the currency by which our bodies are alive. All right, so we phosphorylated this. It's not just because ATP is here, we phosphorylated something. Okay, so now, once this is phosphorylated, we can actually kick this along. This is when we get started, okay, and it will start creating an RNA molecule. And parts of the pre-initiation complex can remain on the promoter to keep attracting additional RNA polymerase molecules. So there are tons of RNA polymerase enzymes present in the nucleus at any one time. And as long as we are ready to start bringing in these pre-initiation complex general transcription factors, we can continue to repeatedly transcribe off this guy, okay? So depending on how available we make this promoter. So again, when we get to chapter 18, we'll see that turning transcription on or off, it's really going to be dependent on how accessible we make this promoter to all of these factors. So if we make it really accessible, we'll get more transcription. If we block access or make it more difficult, we're going to get less transcription. So that's going to be at the heart of gene expression. Okay, particularly we're talking directly about RNA. So we've talked a little bit about elongation. Okay, previously we looked at using the template and the non-template strands, what's going on there. All right, so that's what's going to happen next. We're going to be creating that RNA molecule. But how is this process terminated? So in prokaryotes, this is a hairpin loop that is accompanied by a polyuracil or poly-U sequence that will cause termination. Okay, so this isn't the clearest image, particularly because everything's in shades of magenta, which isn't really a real color, but that's a Google for another time. Okay, so messenger RNA, when we form a hairpin loop, we're thinking about a hairpin being like a bobby pin. So it's going to fold back on itself. Okay, and these are called hairpin loops. We'll see more of these in chapter 18. These are a very common RNA structure. All right, so they're going to just go ahead and fold back on themselves. Okay, and then also we've got this poly-U uracils, chain of uracils here. This is going to cause termination. Okay, for whatever reason, RNA polymerase biochemically not interested in this, so it's going to release. There are also some alternatives to this. RNA sequence combined a protein called Rho that causes release. It's really not important to have to know for every gene what exactly is happening here, but it's important to know that the polymerase in general knows where to stop. So it's got some sequence things going on that tell it, okay, you're done here now. Because otherwise you would just keep going down the entire chromosome. Okay, and in prokaryotes, that's a circle. So you'd be there a long time, man. Okay, so eukaryotes, the RNA has cleaved a few, maybe 10 to 35 nucleotides downstream of what we call a polyadenylation sequence. Okay, again, the important thing here is to know that there's going to be a sequence that tells transcription to finish up. Okay, so once we finish transcription, we know in prokaryotic cells, we're probably already translating by now. We've already made a protein because we're doing that simultaneously. In eukaryotic cells, we have a couple more steps. Okay, so again, only in eukaryotic cells do we have RNA processing. All right, so there are three things, only two are shown here, but three total things that we're going to be looking at. So the first is what we call a 5' cap. Okay, and it is so called because it goes onto our 5' region. Okay, we're also going to have a 3' or poly A tail. Okay, so this is possible because transcription is going to happen in the nucleus separate from translation. Okay, so because we can separate that process out, we have time and need to add a cap and a poly A tail. So the cap and tail will generally help protect the RNA from nucleases. So here's our 5' cap, and just for fun, I've shown kind of the actual structure. So this is our 5' end, okay, 5' carbon, that's what we named it after. Here we go, that's the beginning. And this is actually a sort of backwards guanine. So this is going to be a guanosine, a 7-methyl guanosine. So you see here's the methyl. Okay, here is the nitrogenous base that makes this a guanosine. Here is our ribose sugar. Okay, and that carbon. See how they're stuck 5' to 5' through this phosphate bridge. You don't have to memorize the structure, but I like to think of it in this way as sort of a nucleotide that's sort of stuck on backwards. Okay, it's not really exactly the case, but it helps me to think about it. Okay, this caps the end, and it's going to help protect from nuclease attacks, so chewing up these RNA sequences, degrading them. And the 5' cap in eukaryotic cells also can aid with ribosome binding later on when we get into the cytosol. The poly A tail can vary in length, okay, from transcript to transcript, and it contributes to the stability. So generally speaking, a longer tail will make this more stable, whereas a shorter tail makes it less stable. And again, transients is a good thing, but you don't want this to be too transient. The cell needs to stick around for the exact right amount of time, because what we'll find when we start talking about translation is that as long as this RNA molecule remains intact, multiple ribosomes can keep kind of trucking along this producing protein. So if you leave this hanging around longer, you get more protein. If you don't want a lot of protein, you'd like this to go away a little more quickly. Okay, so the stability is controlled heavily by the cell, and we'll talk more about this in chapter 18. So when we finish up with our RNA processing, we of course have to talk about splicing. So splicing is going to happen in our primary RNA transcript. So this is exactly what we've just pumped out of our RNA polymerase. And you can see that there are exons and introns that are kind of alternating here. So exons are going to be what we keep in the sequence. So that sounds weird because you think X means out, okay? In this case, think of it like EX for expressed. So this would be the expressed part. Okay, it makes it even worse that the part we take out, we've labeled intron. I kind of feel like they should be labeled outrons. Okay, but I didn't name these. The introns will be removed, the exons will stay. Okay, so the expressed regions. We're going to talk about how splicing happens, but let's kind of get a spoiler for the end result. So once we remove these introns, exon 1 will be attached to exon 2 as if the intron never existed. Similarly with 2 and 3. Okay, so I want to take a second to talk about this mature RNA message. Okay, we also have some areas at the 5' and 3' end that we call untranslated. Okay, so we'll learn when we get to translation. There's like a little bit of sequence. It can also be called leader, okay, sequence, but 5' untranslated, or 5' UTR for untranslated. We're going to find that the ribosome is not going to actually translate this. It'll start translation with this exon, and it will finish up here. And so we have a little bit of stuff at the end that's untranslated as well. So actually won't be part of the finished protein, but it's part of the structure of the mature RNA with the introns removed and everything. So let's take a second just to talk about the last part of this video, how splicing occurs. So we're going to go into what I would call a medium amount of depth for this process. So let's start with our pre-mRNA. So we've not removed our introns yet. Okay, and let's look a little bit more closely at this. So let's take one of these introns. So let's take this first intron here, and that's what we're looking at. Okay, you can see exon 1 and exon 2 on either side. In this image, the lighter brown is our intron. So there are a couple of sequence pieces that we have to actually look at here. So one is right at the left-hand border of this intron, we have a GU, okay, guanine and a uracil. About 80% further down, we have what's called a branch point A, or branch point adenine. Okay, keep that in your mind for now. And then at the very last part of the splice site, right before we get to the second exon, we have an adenine and a guanine that are going to be an AG here. And of course, there's sequence between. Okay, it's not just 5 nucleotides, it's a lot of sequence between, but we're going to have branch point A and some specific sequence on either side. All right, so that's what's actually happening here, right? So what we're going to see happen next is that either side of this intron, okay, the GU, okay, to the 3' end of this, we're going to be bringing these sides close together. And this is going to be achieved through the action of what's called a spliceosome. So the spliceosome is composed, you can see kind of the innards here. They're not named, but that'll probably make it a little bit easier for now. Various proteins called small nuclear, because they're small and in the nucleus, ribonucleoproteins, ribonucleo, that's like RNA proteins. So they are proteins that contain some RNA. Yes, there's some RNA actually embedded in them. Okay, not mRNA, different kind. Okay, these ribonucleoproteins, RNA plus proteins together. They're small and they live in the nucleus. Okay, so that makes sense. They're small nuclear ribonucleoproteins. And they are specific, they have specific names that bind this intron sequence and help bring these two pieces together. Okay, so one of these small nuclear ribonucleoproteins binds on the 5' ends where this GU is. Okay, so one of these guys binding here. Okay, the other binds further along this sequence, including this branch point A. Okay, so including the branch point A. So that's what you can think going on here, the GU and a branch point A. So let's see what happens next. Okay, so we have, again, our sequences here, right? And we know we've got our spliceosome ready to go. The proteins assemble and they help bring the opposite sides together. The last part of that is that we're going to cut the 5' ends and covalently attach it to that branch point A that was somewhere over here. Okay, so GU to the branch point A. Okay, this is going to form what we refer to as a lariat. Okay, and we're going to then cleave that AG. Okay, we're going to cleave the 3' end, and what's released is literally a lariat of the intron. Okay, so let's look at the breakdown of this. This would be like the GU right here attached to that branch point A covalently. They are together forever now. We've just cleaved that 3' AG to release this, okay? And then we have some exon joining complexes that will join covalently exon 1 and 2 together here, okay? So the exons will be spliced together. So let's take a second to just look at a visual, an animation of how this happens.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
Okay, so we've taken a moment to kind of visualize how the splicing happens. So while the biochemistry of that is really interesting, the spliceosome is a really interesting complex. The kind of big take home from alternative splicing is really how much diversity it can add to our genome. So it turns out that we don't always have that many more genes than any other organism, but because we're able to undergo splicing, we can create a lot more products from what we do have. So we can check out this idea of different types of splicing. So what we have in this column of pre-mRNAs, this is going to be completely the same transcript. So the same thing that was derived from their DNA. We made a copy of it that's RNA, but different ways that we can splice it and join exons together. So you don't have to worry about the exact terms necessarily. They're just descriptive of what's happened. They're not necessarily terms you have to memorize, but you can see, like in some cases, we might take out 2 separate introns and leave a green exon. Alternatively, we can treat the 5' end of this first intron and the 3' end of the second intron as one large intron and completely remove that green section. We can also have alternative splice sites on the 5' end. Instead of going all the way over here, we can go in a little bit. Instead of on the 3' end, going all the way against this green section, we can move in a little bit. And you can see how the flexibility of that would be possible, because AG and GU, these sequences that we use, they're very common. There's only 4 nucleotides, so you can imagine them happening pretty frequently. So we do have a little bit of play with that. Okay, and you can see, depending on what happens, we're able to add in some extra exon here, okay, from one side or the other of our intron. Okay, sometimes we can leave the alleged intron in, all right, and sometimes we can have, this is a little bit different transcript, sometimes we can have a situation where we can take either one exon or the other. So either I leave the green one in and take the blue one out, or I leave the blue one in and take the green one out, okay? And this can really change the function of the resulting protein, as you can imagine. So the nucleotides that will be present there, we may have extra nucleotides, we may change out a domain on a protein, okay? We can sometimes render something non-functional. And this sounds like it would be just like a crazy thing to happen, like it would happen once in a while, but the vast majority of your genes have at least some alternative splicing, and almost all of them undergo splicing to a degree, but there are also a lot of alternatives that can happen out there. Okay, so once we finish with our splicing, we can have a mature messenger RNA. So again, we've got our 5' cap. We have spliced everything out. Here you can see our leader and trailer. That's our untranslated regions we discussed previously, and our poly A tail. So at this point, we're still in the nucleus. When we get to our next video, we'll export our RNA through a nuclear pore into the cytosol for our eukaryotic cells, and we'll begin the process of translation.